Chapter 16, Elements of Adaptive Immunity. There are quite a few learning objectives for this topic, so hold on. Contrast the flow of lymph with the flow of blood. Describe the primary and secondary organs of the lymphatic system. Describe the importance of red bone marrow, the thymus, lymph nodes, and other lymphoid tissues. Identify the characteristics of antigens that stimulate effective immune responses. Distinguish among exogenic antigens, endogenous antigens, and autoantigens. Describe the characteristics of B lymphocytes that furnishes them specificity. Describe the basic structure of an immunoglobulin molecule. Contrast the structure and function of the 5 classes of immunoglobulins. Describe the importance of the thymus to the development of T lymphocytes. Describe the basic characteristics common to T lymphocytes. Compare and contrast 3 types of T cells. Describe apoptosis and explain its role in lymphocyte editing by clonal deletion. Compare and contrast clonal deletion of T cells and clonal deletion of B cells. List the 5 types of cytokines. In this topic, we're going to be covering tissues and organs of the lymphatic system, antigens, B lymphocytes, also called B cells, and antibodies, T lymphocytes, or T cells, clonal deletion, and immune response cytokines. Let's go over the tissues and organs of the lymphatic system, as well as the movement of lymph through the body. Lymph basically arises from fluid that has leaked out of vessels into the tissues. Basically, it's plasma, but when it's in the tissues, we call it intercellular fluid. When it leaks back into lymphatic vessels, we call it lymph. And lymph vessels carry the lymph from the various tissues back to where it dumps back into the circulatory system near the heart. Now, the body takes the opportunity to screen lymph for foreign molecules. It's been out among all of the cells, and it's a perfect medium for carrying foreign antigens. First off, the primary lymphatic organs are bone marrow and the thymus, which are involved in producing lymphocytes. The bone marrow is where the B cells arise and then they mature in the lymph nodes, and then the thymus is where T cells mature. The thymus is located right here in the same vicinity as the heart. The rest of these secondary lymphatic organs and tissues are involved with screening the lymph. So that would include the tonsils, various lymph nodes that are found in the throat, the armpit, and in the groin. We also have what are called mucosa-associated lymphoid tissues, or MALT, that are found in the intestines. And we also have the spleen, etc. So all of these tissues have an abundance of lymphocytes, and they're looking for antigens. They're looking for evidences of foreign invaders into the body, or in the case of MALT, into the GI tract. So what are antigens? Antigens are any molecule that will stimulate an adaptive immune response. So we have all sorts of properties of molecules that make them good antigens or good immunogens, or poor antigens or poor immunogens. If they have a complex shape, then they're going to stimulate the adaptive immune system. They are good antigens. Their size, generally speaking, if you're too small, you're not going to stimulate the immune response. So we look for big stuff, and we're looking for not only a rigid shape, but complexity in that shape. So let's take a look at this diagram and take a look at what makes a good, well, immunogen is another term for antigen. I prefer antigen. But proteins generally make very good antigens. And not only that, but different regions on the protein, which we call epitopes, are what stimulates the specific immune system. So we're going to find proteins in foreign human or animal cells, microbes, and unfortunately plant molecules that aren't doing us any harm. Things that are poor antigens are small, like triglycerides. It's very, very small, very hard for your immune system to spot, so to speak. Polypeptides have a tendency to be poor immunogens too. Also, you notice that this, particularly with the small carbohydrates, very similar repeating units. There's not a whole lot of complexity for your immune system to recognize. There's not a whole lot of epitopes. So these are the things that lead to a molecule stimulating the immune system or being an antigen. We group antigens into 3 categories. First, there are exogenous antigens. These are things that are outside of a pathogen. They're toxins that are secreted, other secretion, or external components of a pathogen, like flagella. Then there are endogenous antigens. These are pathogenic products that are displayed on infected cells. So if I get a virus inside of a cell, as that cell makes viral products, it's going to display it on its outside. And we consider that to be endogenous antigens because they arose from the inside of the host cell. And then lastly, we have autoantigens, or they're also called self-antigens. These are antigenic molecules that are found on an individual's normal cells. So as we go through, we're going to talk about how your immune system recognizes your autoantigens and ignores them, but will attack them from, say, your brother or your sisters. If you were to receive a skin graft or, say, a kidney in a kidney transplant, your immune system is going to recognize somebody else's autoantigens because they're not your autoantigens. Next up, B cells and their products, antibodies. We're going to talk about the specificity of the B cell receptor, or BCR. We're going to talk about specificity and antibody structure, antibody function, and classes of antibodies. On the surface of B cells are what is called the B cell receptor, or the BCR. Basically, this is the type of antibody that it is capable of making. So it has a transmembrane portion that sticks it into the cytoplasmic membrane. We have this portion that attaches to that. And then we have the arms of the Y, if you want to think of it that way. And on the end, we have the variable region. We call this the variable region because in all humans, this is pretty much the same from person to person. But this variable region not only varies from person to person, but it varies from B cell to B cell. So what happens is, within the cell, genes are shuffled. Everybody's born with a certain number of genes that code for this variable region. And within each B cell, as they're developing in the bone marrow, they produce a different order that each leads to a different variable region. So what they do is after they are produced in the bone marrow, they go to the lymph nodes and they hang out with their BCRs waiting for an epitope that will match their antigen binding sites. Once that happens, then they go on to become fully mature B cells. And we're going to talk about that more later. But keep in mind that these receptors vary per B cell receptor. Now this antigen can be either presented by an antigen presenting cell, that's usually what works best, or it can be a soluble antigen that's floating by in the lymph. In either case, once it binds, then the B cell will start dividing and produce more of itself. Now the whole point of dividing is so that you get more cells that have that BCR, and they mature into either plasma cells or memory cells. Plasma cells, their whole job is to pump out immunoglobulins that match their BCR. Memory cells kind of hang back and they're ready for the next time that antigen comes in contact with its BCR. So let's talk about the structure of immunoglobulins, or antibodies. Here is more of a molecular formula that you can see some beta pleated sheets and some alpha helixes. This is more of a simplified drawing. This is what your basic antibody looks like. We have the heavy chain and the light chains that are connected by disulfide bonds, which makes antibodies very stable. And this part of the heavy chains and this part of the light chains contribute to the antigen binding sites, or the variable region. This binds the same epitope that the BCR variable region bound. This makes the antibody very, very specific for a particular antigen, or more specifically, a particular epitope on the antigen. Now lastly, down here on the end of the heavy chains, that's opposite from the antigen binding sites, is what's called the Fc portion. This becomes important in some of the functions of antibodies, of which we're going to talk about now. First off, antibodies can neutralize pathogens, viruses, or toxins. So if this antibody is specific for an adhesion protein on either a bacterium or a virus, they can cover that up and the bacterium and the virus can't attach to their host cell. It neutralizes them. Now, it can also attach to a toxin and do the same thing, covering up an active site, covering up the effective portion of the toxin that causes whatever effect the toxin has. It neutralizes it. Now, once these are neutralized, they can be engulfed by phagocytes. Antibodies act as opsons. Phagocytes have receptor proteins for the Fc portion of antibodies. So they grab a hold of this part. It makes it easier for them to adhere to the pathogens, and they swallow them on down. Now, agglutination is a fancy word for clumping. When you get an antibody that attaches to an antigen on two different cells, it clumps them. Now you're thinking, why in the world would that be a benefit? Well, if they're clumped, they have a harder time moving deeper into tissues. They have a harder time of spreading. Also, when the phagocyte comes to swallow them down, it can swallow down all 6 of these cells instead of having to chase them individually. So it aids opsonization by clumping them. And then there's antibody-dependent cellular cytotoxicity, or ADCC. Basically, we get antibodies attaching to their epitope, to their antigen. Natural killer lymphocytes, NK lymphocytes, also have an Fc receptor protein. They bind to that, and they know if there's any place where antibodies are attaching, that's a problem. So they attach, they release porphyrin, which creates a hole in the target cell and causes it to lyse. Now for 2 functions that are not on here, but which we talked about in the last chapter. Antibodies are also important in the activation of complement. Remember, that would be the classic pathway. And also they are mediators of inflammation. Now on to the classes or types of antibody. First off is IgM. IgM is a pentamer. See, we've got 5 antibody molecules that are joined by a J chain, and it is the first antibody that is released during a primary infection, or in other words, during the first time that your body is exposed to a particular epitope. IgM are very good at agglutination. As you can see, they've got 10 hands, so to speak, that they can grab onto 10 different cells or viruses. But it's expensive to make. As you can see, it's just a big, scary molecule. So, during the primary infection, plasma cells that are making IgM will switch to making IgG. We call this class switching. IgG is a monomer. It's just one antibody molecule. It's not as good at agglutination as IgM, but it's a whole lot less expensive for the body to make, and it still does all of the jobs of an antibody nearly as well as IgM. So if you are to look at the blood and look at the various levels of immunoglobulins, you're going to find more IgG in the blood than anything else. Also, because it's a smaller molecule than IgM, IgG can cross the placenta. So a mother's antibodies can cross the placenta to a fetus and protect the fetus and newborn children from various pathogens while they're still developing their own antibodies, while they're being exposed to antigens. Now, sometimes a cell that is making IgG will class switch again, and it will either switch to making IgA or IgE, which is also a monomer. IgA is a dimer, and it's also connected by a J chain, just not as extensive as for the IgM. IgA is found in secretions. It's found in mucus, it's found in breast milk, it's found in tears. Any type of secretion, saliva, you're going to find IgA. So IgA protects your mucosa, and it also adds to the maternal protection that babies can receive if they're being fed breast milk. IgE has some good functions, but we're mainly going to focus on its bad functions, which is its involvement in allergies. Cells that are producing IgG will class switch to producing IgE, which has a tendency to attach to mast cells or basophils. So they're hanging out when they contact their antigen. Specifically, if it's an antigen that is involved in allergies, we call it an allergen. When IgG binds its allergen, it will stimulate the mast cells to release large amounts of histamines, which triggers inflammation and the annoying symptoms of allergies. Treatments to help resolve allergies are involved in getting these cells that produce IgE to class switch back to making IgG, which doesn't attach to basophils and mast cells. And IgD is the antibody that's basically soluble B cell receptors. Sometimes the receptors come off of the B cells and we find them floating around in the serum. Not very often, though.